Thank you, Marco. Good morning, everyone. First of all, I would like to say thank you for this beautiful opportunity to speak here, because Professor Marco Cosentino was thinking that actually speaking about some real technique for this school would be very interesting, especially for understand the importance as these techniques as a very, very fundamental tool in research. It's actually a great opportunity. I'm a technician, so for me actually it's the very first time that I speak for something like that with the audience, and I was questioning myself. Okay, in these days we had the good fortune to assist to very elegant presentation from some great international professors. And so the first question that I was asking to myself was, why am I here? Okay, because I'm a technician. And why? I would like to explain to you that it's very difficult for me try to be interesting and not to bother you with some too much technical question about this technique. Why am I here? I would like to start before Professor Cosentino was speaking about the knowledge about technique, because actually knowledge is consciousness, and consciousness is understanding the results that you can handle actually, and if you know what are you handling, actually you can use in the proper way. So this is the reason because I'm here, because I would like to give you some suggestion for understand how can be will be useful to understand properly the techniques. Also, Manu before was speaking about flow cytometry, very good, in very good way, very technical way. It's very difficult also to understand deeply this technique. And I would like not to go very deep in this presentation, but just to give you a very light presentation for understanding the method, and for understanding how you can use in a proper way the results from a real-time PCR. Okay, yeah, Hollywood effect, this is great. First of all, look at this guy, beautiful, sweet James Bond suite, it's beautiful, this guy. This actually, this guy is the origin of all our disgrace, because this is Kerry Bank-Mallis. He's actually the inventor of the modern technique of PCR, and he is a Nobel Prize winner in the 1993 for chemistry, and actually he was a Californian blonde surfer. Okay, he himself was saying that when he invented PCR, he was under drug effects. Okay, actually it's incredible how this brilliant mind actually divide the modern science in pre-PCR and post-PCR era. And it's incredible for me, because sometimes when people speak to me and say, okay, you are a joker, you all time are not serious, when I look at this picture, I think, okay, maybe I'm in the right place. Okay, he was actually brilliant, using actually some very simple chemistry that was already known in biology. And basically, he developed the polymerase chain reaction for practical use in PCR. So, for understanding what is real-time PCR, that is actually the technique that I will speak today, it's better to start with some basics knowledge about the PCR old-fashioned technique. Yeah, PCR is a technique for amplifying DNA. Okay, there are two reasons for amplify the DNA. Basically, you may want to simply create multiple copies of a rare piece of DNA. For example, forensic scientists like CSI, for understand, may want to amplify a teeny piece of DNA from a crime scene. Okay, so you can amplify the proof. Or more commonly, however, you may wish to compare two different samples of DNA to see which is the more abundant. Okay, basically why you amplify? Because you need a strong signal, something big. You cannot look like Orazio in CSI that is looking at the Eppendorf and immediately, yes, there is DNA. Okay, you need amplification for do that. The responsible of this amplification actually is a thermostable polymerase enzyme, drives the PCR, and synthesize complementary sequence of bases to any single strand of DNA, providing it has a double-stranded starting point. It's very useful, this, because you can choose which gene you want to amplify. Just adding small pieces of DNA complementary to this gene of interest. These small pieces of DNA are known as primer, because they prime the DNA sample ready for the polymerase action. Basically, during the PCR, the chain reaction, changes in temperature are used to control the activity of polymerase and the binding of primers. I know that I'm boring now for this, but trust me, repetita adjuvant, say Latin, so I will go very fast in this, not to stay too much. The beginning of the reaction is at 95 degrees. Today is not like that. Today we have a hot start technique that can start also at 80 degree. Okay, 95 actually is the golden standard, the standard rule. Okay, so at 95 degrees, the double-strand DNA actually is melted in two single strands. Then the temperature is then lowered to 60 degrees, and this also allows to the primer to bind to your gene of interest. You can see the two little pieces of the two primer that can finally bind. Then the polymerase, at this point, it can just start it can begin to copy the DNA strand. So as far the reaction go on, you have actually the duplication of this strand. The optimal temperature for the polymerase to operate is 72 degrees. So at this point, the temperature is sometimes raised to 72 to allow the enzyme to work faster. There are now when the polymerase ends the first cycle, you have now actually twice as many copies of your gene of interest as when you started. This temperature change is repeated through around 40 cycles. So for 40 times, you just raise the temperature, lower the temperature, up and down, up and down. Like this, you just build DNA, and then you splice in two, and then rebuild, and so on. After amplifying your gene, it is possible to run the amplified DNA out on an agarose gel. This is an old-fashioned technique of PCR. And stain it with a dye which make it visible, so something that is markable. The brighter the visible band, the more copies of your target you have created. Now, this is the first point in which you must put your attention, because now we have a lot of works that are based on old-fashioned real-time, old-fashioned PCR. So at the end of the reaction, you have this agarose gel, and you have this visible band in which you understand how much of your gene of interest you have. But you will see in following in this presentation that this is not actually correct, because if you are speaking about yes or not about some gene, is okay. If you are speaking about quantity of this specific gene, the old-fashioned PCR is not the best approach. And I would like to say, pay attention all the time when you are reading some paper in which you have just results in normal PCR. Because today it's sure that it is just a quality approach, not a quantity approach. On the same principle, okay, the same principle of amplification is employed in real-time PCR. The difference is then within the real-time PCR, actually you can see the reaction of amplification of DNA during the process. As you can see here, this is a typical fluorescence signal that provides you the data about the well function of this reaction. As the number of gene copies increase during the reaction, fluorescence increase. For do this, you need to mark your amplification, your template with some fluorescence. Real-time PCR has many benefits over the old-fashioned approach. Firstly, give you a look into the reaction. Basically, you can see in each well if the reaction is going good, is going well. And this is very important because, you know, in the old-fashioned, until the end, you cannot see nothing, basically, and it's like a step of the faith. You must pray for God if something is good. Until the end, you cannot see nothing. Actually, also with the real time, you can do nothing. Okay, but at least you can understand before, so you can immediately redo another experiment, for example. The efficiency of the reaction can be and must be precisely calculated. Must be, and we will speak later why. It's very important. There is also no need to run the PCR product out on a gel after the reaction, so time of experiment is it's shorter, it's much shorter. And the greatest advantage for all over, however, is that real-time PCR data can be used to perform truly quantitative analysis of gene expression. In comparison, old-fashioned PCR was only ever semi-quantitative at best. Okay. So, real-time, again, is an exceptionally powerful research tool, and today is very easy to have in your hand very good kits. I remember when I started, after I started performing real-time PCR around 2005, 2006. And I remember hour with Professor Ferrari stay on the desk and, for example, with this primer design, searching for every possible solution, etc., 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 etc. Sorry. You noticed that I'm Italian, maybe. Okay, and I remember that. Now, today is much more easier, but in my opinion is also very, very dangerous, this kind of approach, very easy. Because the trend is not to think, what are you doing? So it's very easy to have the right stuff in your hand, but also it's very easy to have the wrong stuff in your hand for performing your assay. In any case, whether you are an expert or a complete beginner today, you have a lot of companies that can make your life very easy with specialists, for example. But especially with this reagent that today you can just put your sample in one Eppendorf, like Orazio in CSI, after half an hour you can have some results. After centrifugation, yeah, is enough, one centrifugation and you have the results. No, actually I would like to say you this thing. It's all about the quality, in my opinion. And today we are actually working, especially in science, we have this trend, everybody is running, okay? And technology is doubling the power, actually. Every 6 months, 3 months, you just start today to do something and tomorrow is old-fashioned stuff. This is very interesting, especially if you have money to spend in research. For example, San Raffaele, okay, with all the economic issues that he has in the last year, but if you go there, if someone of you is coming from there, you can see one of the best laboratories for biochemistry and molecular biology, for example, in San Raffaele. And I had the good fortune to work there for some seminars, etc., etc. Sorry. And this is very, very, it's beautiful because you feel the power of the research. Okay, you have these big instruments, very fast, very accurate. But then you understand that, for example, Eppendorf itself, when was testing some new products, go to San Raffaele, but also come to our center of research, because I think to myself like an artisan. For me, less is more, if less is good. Okay, and this is philosophy. So when you are running, and you need to do everything faster and faster, it's the first mistake that you can do is not to think what are you doing. And this is very dangerous. When I was speaking with Marco Cosentino, we have a very short briefing about this, this lecture in this seminar, and I was saying, okay, Marco, what about my intervention? And he was like, okay, you know, we have in program that in our center of research we will do some workshop. This is not a workshop. Workshop is when you work, okay? And in the program, in the future, we hope, if we have the good fortune to have also money, but the program is to have some real workshop, and for go deep in all of this. But I was asking, say, what about my lecture? He just say, okay, this must be like a teaser trailer, okay? I would like that after this speech, you will have a lot of questions, a lot of, okay, why this, why this, why this? So we can go deep in future about this, okay? And continue. What you need for do real-time PCR, actually? You need this monster machine? No, actually you need just a thermocycler that is actually not cheap, but also not very expensive machine. We are speaking about around from 5,000 until, okay, you can go until 50,000 euro, okay. But with 5,000 euro, you have sufficient skill for perform your assay. Then you need kits, extraction, and a lot of stuff that you can buy, as I was saying before, with a lot of companies. Okay. I was speaking about primer design before. Primer design is this software that can allow you to project actually your primers for your interest gene. But today, actually, it's very, very simple to find beautiful assay for whatever you need, actually. Today, also, good companies allow you all the technical results, validation results of every single primer. So actually, is a lot of work less respect to the past. So what you need and what we have actually in our Center of Research in Pharmacology. You need organization of the space. Why? Because all about good results in real-time PCR is about a contamination or degradation of your sample. So it's important to understand that the ambient where you work, it's fundamental for having good results. Organization of the space is in this mean actually you can have separate spaces for different technique, or just organize very well a single lab for have beautiful results. Also, the workflow is very important in real-time PCR. We will see this later because cross-contamination is one of the most important danger in this technique. And all of this, all of procedure that you apply for this assay, all of this bring you to a quality work. But this is not enough. You need also quality check of your work. For example, I like so much working blind. You know, several times happens in our center of research that someone that is interested in searching gene expression send us sample. These sample for me are just cells. I know that the kind of cells, and they ask me, okay, search this kind of this gene after this. I don't know treatment. I don't know nothing basically. When you can work like that, when you are sure that your results are good. Because you cannot work in blind if you are just use the dice. No, you cannot. So it's important that your quality check provide you the skills for have strong results. Important. Okay, always. It's important safety for operator. We are not speaking about Ebola when we are speaking about safety. Simply this strand, the DNA strand, etc., or also especially the chemicals in the kit are very dangerous. So it's important that in your organization you think about that. Safety for the samples, of course, because without good samples you cannot have good results, and accuracy and efficiency of results. Okay. Speaking about contamination, we are speaking about one of the two most important enemies in real-time PCR. Contamination, in our case, is not about this guy. Okay, this is for Alessandra Luini, my colleague, because when we speak about contamination, it's not microbiological contamination. So not this, but this. This is, you know, if you watch the movie Jurassic Park, this was the DNA from Jurassic Park. Because actually the product of real-time PCR or PCR is the most important contaminator for our experiment. So this is important, and it's very important to understand that you have always a pre-amplification zone and a post-amplification zone in your lab. It's not important that these zones are physically in two rooms separated, but, for example, your procedure, your workflow must consider this aspect. So, for example, in our lab, we have a big room for molecular biology, a big room, but actually we avoid contamination by several steps of UV irradiation, for example, in the ambient. So we use laminar flow with UV, and we sterilize all the time the ambient after each passage. Ecco, so you can adopt a couple of strategies for this. I was speaking about the workflow in molecular lab. Okay, first of all, you are preparing your reactions, so you will use a lot of reagents and kits that are actually, depending by the technique that you use, for example, for extraction of RNA, they can be very, very practical and very, very user-friendly, or very difficult. For example, the old-fashioned chloroform technique for extracting mRNA. So this is a red zone. Why? Because when I'm preparing my mix, for example, for amplifying, it's very dangerous if some reagents go in some well, or, for example, the cross-contamination between the reagents. So it's very red zone, this. It must be very accurate and precise in handling, actually, this. Then we go into this yellow zone, because during the extraction, actually, you don't have big problems, exception for your handling. You must be very expert for not to contaminate yourself the extraction of the samples. Then we are in this green zone. It's totally safe. Why? Because after your preparation, even if you are in the first step of reverse transcription or in real-time PCR, you have actually your sample in the thermocycle. So there is not risk of contamination there, because the sample cannot jump out, especially if you cover this. They can jump out if you don't, for example, cover with the cap and you don't have good thermocycler. But actually it's a green zone. Then again, the post-amplification area, especially after reverse transcription, it's very dangerous, because when you open, for example, put in in real-time PCR, when you open your cDNA, it's beautiful to see sometimes this aerosol that is going in the ambient. And this is the most dangerous step in your workflow. Anyway, the workflow is one-way workflow. After this, or you change your table, for example, and your ambient, your room, or actually you need to do some UV decontamination. Go on. Okay, the most important contamination are carryover, when you are handling sample and you just, okay, forgot to change the tip, and you just do a big mistake. Then exogenous DNA, in our hand, in everywhere basically there is exogenous DNA, especially in your sample. In your sample you have a lot of exogenous DNA, so if your purification of RNA is not good, you can have a cross reaction with the endogenous DNA, so false positivity. Cross-contamination, the same, basically, by samples. In all these cases, you have a risk of false positivity. So you can amplify something that is not for your interest. The second enemy that I don't put here because actually it's a very long and practical speech is the degradation of mRNA in this case, or DNA. Why? Because if you are performing something with DNA, like Professor Ferrari will say in the next presentation, DNA is very stable. Remember Jurassic Park? It's very funny. It's a movie, okay? But they are, okay, it's fantascientific, but they are actually taking DNA from old mosquitoes in this rock of cos'era? Amber, okay? It's possible because DNA is stable for years, centuries actually. mRNA, no. mRNA, if you look at him too much longer, he just go, it's finished. So degradation of mRNA is the second very important aspect to preserve, our samples. How to preserve this? I can say now the first teaser. You will see this in our workshop if you want. Actually, important is how to put mRNA in storage, and actually how to proceed how to handle the sample during the entire procedure. For contamination, but also for degradation of your samples, actually, the prevention is good laboratory practice, always. I don't know how many of you actually are involved in practical experiment in laboratory. I see a lot of young researchers. I think that most of you are in lab. So how to handle the sample, how to move in lab is the first rule, always, okay. Experience, of course, and we will see later that actually the good results when you don't have these beautiful, fantascientific big machines, the beauty actually of your results become only from your hand. So it's important to have a good experience, a long experience. And then constant formation for operators. For example, coming in workshop, going in workshop, okay? I'm good to tease you. Okay. So actually, what is now we enter much more specifically in our technique. We will speak about real-time PCR, and we will have some very short, but I hope good example of gene expression. That is actually my field of interest. Wow. Okay, real-time PCR, also called quantitative PCR or qPCR, can provide a simple and elegant method for determining the amount of a target sequence or gene that is present in a sample. Its very simplicity can sometimes lead to problems of overlooking some of critical factors that make it work. Okay, now we will enter in the terminology of this technique. Okay, this is always the classic diagram of a sample in PCR. There are few words that you need to understand and remember. First of all, threshold. It's a level of delta Rn. No, maybe it's better to start from Rn. Is a normalized reporter, is the ratio of the fluorescence emission intensity of the reported dye to the fluorescence emission intensity of the passive reference dye, ROX. What I'm saying, I don't know. No, I will explain to you. Basically, in every instrument, you have always a passive fluorescence. Also in flow cytometry, we see before that you have autofluorescence at some level. This can be measured by the instrument all the time by this passive signal that is ROX, actually. In every single step of the reaction, you have this passive fluorescence that is more or less constant, exception for the aspecific fluorescence that is coming cycle by cycle in your reaction. The machine provides to give you the ratio of this. So basically, Rn is the final results of your zero line, is the fluorescence that is actually your background signal. Is different from the threshold, because the threshold is a level of delta Rn used for the determination of the threshold cycle in real-time assays. The level is set above the baseline, but sufficiently low to be within the linear growth region of the amplification curve. The threshold is the line whose intersection with the amplification plot defines the CT. You see in the picture, the threshold is this uncontinuous line, and actually is not a baseline, is the line that is above the noise that you don't need. And when your reaction is growing up, your fluorescence is increasing. And when this signal is enough strength to exit from the basic line, you have the actually the linear grow region in the amplification curve. In this point, you have the CT, actually the point where the fluorescence exits from this line. Baseline, okay, is not so important, is the initial cycle of PCR, it is this found, and threshold cycle is very important because it is exactly the cross, the cycle number at which the fluorescence passes the threshold. Okay, when the fluorescence is enough strength, she can pass. Okay, now coming back to the start, when I was speaking about old-fashioned PCR, this is a great example. It's basically, you can see this in all the presentation in real-time PCR. Now it's 15 years that I see this picture all the time. But basically, it's very nice because you have 96 replicate samples. Replicate means that you put the same quantity in 96 well, and you perform at the same time the same reaction. In real-time PCR, you expect what you expect. You expect that you have the same quantity of the target that you are looking for. And this is exactly what happened here. Around the 26, if I see correct, cycle, you have all the samples express the same quantity of the gene. Then you go until the end. We were speaking about around 40 cycles. At the end, you run the gel. When you run the gel, this is the typical result of good result, actually beautiful gel of this a part of this 96. But look, it's very clear that if you just have this point of view, you think that they are not the same. They are not the same. Is it correct? No. If my experiment was set correctly at the beginning, I expect that they must be the same. This is the reason because old-fashioned style PCR is just for a quality approach about present absence. Now, please don't trust to all these papers that are speaking about PCR, old PCR like quantity method, quantitative method. Sorry, thank you. Okay. But how to show these results? You must use some chemicals for a heavy signal. Basically, the two most common chemistry are intercalating dyes and Tachman probes. Intercalating dyes, for example, cyber green, for speak practically, the principle is intercalating dyes are small molecules that can bind the double strand of DNA. These molecules can emit fluorescence only if bound to DNA, for example, PCR product. Emitted fluorescence is proportional to amount of DNA available. Basically, with these dyes, anything amplified is signal. Intercalating dyes can generate signal if the amplification is not specific to. This is less a problem today. Was a very big problem with the first generation of cyber green, for example, dyes. Today is much more accurate and easy to have good results even with this kind of intercalating dyes. In any case, at the end of your amplification, it's better to check specificity of product with the melting curve. This is a good melting curve, for example. When you have at the same peak, all your amplification is going down, basically you know that you have a specific product. If you have a lot of peak, it's not a good sign. Actually, the most accurate and also difficult to use technique is the binding with Tachman chemistry. The principle is FRET. Effect is used to keep the signal silent. 5 nucleasic activity of Taq DNA polymerase is used to generate the signal. The fluorescence emitted is proportional to amount of DNA available. And real-time PCR with Tachman chemistry is a normal PCR plus the Tachman probe. The principle of FRET is this, basically, forced resonance energy transmission. You have 2 chromophores, one donor and one acceptor. Energy transfer from donor to acceptor when they are in proximity, less than 10 nanometers. And when the distance increases, no energy transfer occurs. So basically, you can have the emission of signal. In Tachman probes, donor is a fluorophore, acceptor is a quencher in proximity, so quenching effect, so you have a quenching effect, so no emission. In distance, you have the fluorescence emission. For be much clear, this is much clearer. Tachman polymerase is able to displace hybridized probe if found during strand replication. So when Tachman is working, basically, if you don't have a specific binding, she can very easily displace actually your primer, basically. If Tachman do the displacement, basically this remain together, and you don't have emission of fluorescence. If the binding basically is very strong, when it's displacing is not possible, Tachman polymerase starts cleavage to 5 ends of the probe 1 nucleotide at a time. This ability is used to separate the fluorophore to quencher, and so you can have finally the emission of fluorescence. In this technique, only probe binding is signals. Only specific binding is a signal, give you a signal. A strong binding of the probe occurs only to specific target DNA. So only specific binding generates signal. Only specific amplification generates signals. In this technique, you can have actually multiplex assays. That I will speak now about this. We are speaking about high specificity. Sorry. If you have specific probes that can have specific fluorescence in different wavelength, you can put different probe in the same well, having in this case a double signal of fluorescence. This is very useful for check, now we will see, in the single well if you have a good amplification, if you have a good reaction. We will go further there. Application of real-time PCR, you see, everything for research is good, okay? Present or absence, absolute quantification, gene expression, microRNA, allelic discrimination, mutation screening, copy number variation, methylation analysis, protein expression interaction analysis. In clinical, there are few, but it's just because you need a very, very specific approach. For example, absolute quantification is mostly used in clinical. For example, when you need to count the load or the virus load in sample. But actually, you can use a real-time PCR for anything you need. The problem is if you really need real-time PCR in your research. So we already speak about the threshold cycle, so this point where the signal exits from the threshold line. Standard curve efficiency of amplification and housekeeping gene are now the argument for the next point. Okay. First of all, threshold cycle, CT. In real-time PCR, as far you go in reaction, less product you have, less copy number of your gene of interest you have, because you need a lot of cycle for have amplification that is enough for be detected. So basically, in this graph, you see, you have 5 lines. These are actually the 5 CT in which the signal exit. This one is the most abundant presence of copy of your gene, because exit first, and as go on, this one is the less present. We are speaking about logarithmic system. When we were speaking about the old PCR technique, basically every cycle corresponds to a duplication of your strand. So from 2 you go, from 1 you go on 2, 2, 4, 4, 8, 8, 16, etc. So this is a logarithmic scale based 2. How have actually a real quantification, absolute quantification of time PCR, as in every other biochemical technique. Basically, you have a standard curve, a dilution series of no template concentration established for determine the initial starting amount of the target template for assessing the reaction efficiency. Basically, you have this standard, must be very accurate the quantification of your standard, and you do just the dilution of this. Doing the dilution, you actually have a standard curve, and when you have your unknown target, you just go on the standard curve and you can have the quantity. It's just the method. This is sorry. Okay, so basically, with my standard curve, I have this point. When I have an unknown target, I just go across to the standard curve and I have the quantity. The standard curve is very important because it is the first step for understand efficiency in real-time PCR. And actually, this is the most important issue that we must consider when we do this technique. Ideally, the efficiency should be the 100%, meaning that template doubles after each cycle during the exponential amplification, as I was saying before, from 1 to 2 to 4. Okay, when this happens, is perfect efficiency. We have 100% efficiency, but we are speaking about not some robotic stuff. We are speaking about a biologic system. In any case, we are speaking about something that can change. So efficiency can be not exactly 100% for your reaction. Okay, this is the slope. So I want that you look at this. Okay, on this column we have the CT number. Okay, the number of cycle, and on this column we have the quantity of DNA that we put in the reaction. Ideally, when we start, we can understand that we put just 1 copy, for example, of DNA. This means that at the first cycle of our reaction, this copy becomes 2. And the second cycle, this becomes 4. Third, 8. At the fourth, 16. The question is, for having 10 copies, at which CT number I can have 10 copies? I think that it's very practically, you can see that it's something between 3 and 4 cycle. Between 3 and 4 cycle, you can have 10 copies of your target. Actually, is it's between 3 and 4, is after 3.32 cycle that you have the tenfold augmentation of your copies. This is very important. I will explain now why. Okay, basically, if your dilution of 10 folds have a distance one from the other of 3.32 cycle, your slope of efficiency is 100%. And this is what I just say. For example, if between one and the other sample in dilution, tenfold dilution, you have less than 3.32 cycle, we are speaking about over efficiency. Why? Because our system requires less time for double your DNA. So if you have less than 3.32 cycle, we are speaking about hyper efficiency. But if, for example, your slope, your difference between the two dilution is more than 3.32, for example, 3.6, you have lower efficiency. And for example, you can see in this case, if the distance between dilution that you made is 3.6 cycle, efficiency is 90%. Why this is important? Because when you put in the same system, for example, two different dyes, two different assay for measuring two different targets in the same sample, it's very important that they have the same efficiency, because if they don't have the same efficiency, you can have over amplification on one target and, for example, lower amplification of the second, giving you something that is actually not the real situation in your sample. Okay. In practical terms, efficiency must be between 90 to 110% for be a good efficiency in one single target. We are not speaking yet about multiplex assay. Okay, how I can verify this? Actually, doing this dilution. Basically, I'm testing all the time when I'm doing some new experiment. I'm testing my probes, doing this dilution and calculating the efficiency. Actually, you can then correct the percentage of efficiency by dilution in your final mix about your reagents. The efficiency should be uniform between target gene and housekeeping gene. What is housekeeping gene? Housekeeping genes are knowing the amount of mRNA in one sample from one specific gene does not tell us a lot. For example, you don't know the total amount of mRNA. Also, you don't know how much the mRNA level has changed compared to the other mRNA levels. We must compare the expression of the target gene to another gene, which expression is normally constant. This is defined normalization through housekeeping gene. What does it mean? Basically, when we are performing real-time PCR, we have, for each target that we have one signal. Okay. This don't give us the exactly amount of DNA. It's some quantity of DNA, but respect to what? We don't know that. Usually, for example, in old-fashioned PCR, you put as control beta-actin, for example, for each sample, and this gives you actually the baseline on which you can compare then the quantity of your target. In real-time PCR, this is much more quick, it's much more easier, because you can, in the same reaction well, you can put two signals, one for housekeeping gene, can be beta-actin, can be 18S rRNA, and for your target. What happens? Happens that if for every single well, you have the first signal that is fixed. It's the same for every sample if you charge the same quantity of DNA, for example. And then you have your target that can be more or less expressed. Okay. We can have a lot of housekeeping genes. why is important to choose the right housekeeping gene? Because they can have some specific reaction to the treatment. Housekeeping gene must be stable in every single condition, in your actually experimental situation. For example, we use 18S rRNA in our lab. That is very complicated to use because it is very abundant in the cells, but actually is totally inert. You don't move with any kind of treatment, or at least treatment that we are using in our lab. So you can have different kind of quantification methods. Absolute quantification, that gives you the exact number of copy, requires that the absolute quantity of the standards, plasmid or in vitro RNA, to be known by some independent means. Basically, you need a known quantity of your standard. And then relative quantification, in my opinion, is much more interesting in research this, because determines the change in expression of a nucleic acid sequence target in a test sample relative to the same sequence in a calibrator sample. The calibrator sample can be an untreated control or a sample at time 0 in a time course study. Relative quantification is commonly used to compare expression levels of wild type with mutated alleles, or the expression levels of a gene in different tissues. In the quantification methods, you have standard curve in absolute quantification and relative quantification. Standard curve in absolute quantification is provided by known specific standards. In relative quantification, standard curve is, for example, done by your internal control in your experiment. Comparative quantification is a mathematical determination. Calibrator sample is used as one pair, actually, base standard. Particular use when particular ratio are expected or to verify trends. I would like to now explain you this later. When you use, or you should use, absolute quantification, in microbiology, for example, for identification and monitoring of pathogens, or in oncology, when you need to monitoring chronic leukemias, for example. In all quality safety testing, measurement of contaminant pathogens, basically all the time that you need is exact count of your copies. Okay, again, standard curve is what we use. You see, standard curve, in this case, if we check the CT in this standard curve, they have around one cycle of difference. When you have standard curves like this one, they are very difficult to set, but actually this is a double-fold dilution. So I dilute all the time half and half and half and half my sample for do this kind of curve. In relative quantification, we use this for gene expression, for Kaplan-Meier analysis, also for microRNA screening analysis. microRNA screening analysis need bigger instrumentation, is much more complex. Okay, again, for efficiency, we have to prepare our standard curve. And actually, relative quantification without housekeeping gene is very difficult, because actually all the time you are providing a different ambient for your reaction. So with this kind of quantification, it's very difficult, for example, to do long-term studies. Okay, this is not important. I will speak all together in this. The most important, most actually efficient and accurate way to speak about results in real-time PCR is when we are speaking about comparative quantification. Comparative quantification actually is a mathematical model that considers this. Basically, in your reaction plate, you have to put all together housekeeping gene that provide your actually your control, internal control, and your target gene. In this I will not explain, or also because for me it's very difficult to explain all of this. But basically, what happened for the properties of the logarithm, for the logarithmic properties, we are just doing the ratio between our target gene to our housekeeping gene. Ratio for the logarithmic properties is like to do a delta, basically. So doing the delta with every single control, you have actually, you set basically your baseline, and you have the amount of your target of interest gene. For be much practical and for to be practical and much clear. This is a typical, this is actually an old diagram of a multiplex assay. Multiplex assay, as you can see, provide us a lot of data. In this case, we consider just this double multiplex, in which we have a signal for housekeeping gene and for example, X gene 1. As you can see, we have in this case 24 replication for this sample. We have the same housekeeping gene that is actually our it's like a baseline for our signal, okay? And then you have the gene of interest that is actually exiting after, for be much clearer, okay? For each one of this sample, we do 3 replicates. Replicate 1 show us the CT of housekeeping gene that is 10.03. And the first gene is exiting at 23 CT. Second replication, housekeeping gene 10.08. Replication 2 of the gene of interest is 23.05. You see how much they are very near. When we do the third replicate, again, housekeeping gene is around 10, and the first gene exit at 23. This 10 and this 23 are the CT cycle, cycle threshold. So basically, the distance between these two genes tell us what? That housekeeping gene is much more expressed of the other respect to the other gene, because it exits after just 10 cycles, and the gene exits around 23. So is much less expressed about housekeeping gene. If we come back on this, okay, several, you see that the gene 2 is exiting around 33, so is less expressed than the gene 1. But you can see that in any case, housekeeping gene have the same expression. So basically, this is like we have set the, okay, our baseline. So which of these two genes is more expressed? Of course, the X gene 1, because it's the first that is exiting respect the 2, and both of them are normalized by the housekeeping gene. And let's go there, forward. Okay, basically there are this mathematical model that is a lot of, seems very complicated, but basically what I'm saying is this. Basically, you must compare every single gene with the, in the same well, housekeeping gene. For doing this, you are doing a ratio that is in concrete like a delta. You do the delta, so you have normalized your interest gene. When we have the results, you have something like this, okay? In triplicates, you have your housekeeping gene, and you check for sure, you must check that they are the same, because you are charging the same quantity in each 3 wells. And also you check the X gene. In this case, this was like the first example that we saw before. So when you look like this at these results, and you see that, okay, this 10, 10, 10. So basically I put all the same quantity, and the amplification is good for all 3, and also for my gene of interest. Then I do what? I do delta CT. Basically, I'm doing the difference from this between this and this, and actually it's just a difference, a delta. This is the distance between the two signals that I have, okay? Then this is actually what sometimes gives some problem, because we are speaking about threshold cycle that are in logarithmic scale. So these are not natural numbers, are number logarithmic. So for transform this result in something that is actually a real number, you must develop the logarithmic number. Actually, we are in a base 2 logarithmic scale, so the number is 2 elevated at the number. Okay, and then when you have all this is okay, and you do the 3 delta CT, you actually do the average, and this is your result. But also, you see, also you can do the vertical average immediately. Okay, you just do the average of all the 3 replication, and at the end the result will not change, it's the same. Okay, now for concluding all this mess about line and target and stuff like that, I will show you just an application, practical application in immunopharmacology, neuroimmunopharmacology, such as this is an expression of gene expression of DRD's receptors in PD patient and controls. Basically, we are speaking in all these days about dopamine and dopamine receptors. This is a typical final graphic in real-time PCR that shows us actually housekeeping gene and all the 5 receptors in in this case, there are 3 different patients or control patients. All these data are performed on CD4 positive cells. And for have this signal, this is housekeeping gene, you remember, we are using the 18S rRNA. You see how they are very the same, because we are actually putting the same quantity of starting material. So this provides us, this is the sign that we are actually working with the same quantity of genomic material. It's multiplex assay, because we are not running one by one all these fluorescence, but at least, sorry, what is this? Okay, at least we are running 2 signals per time. It's like, okay, housekeeping gene, and for example, this is DRD2. You see, in this replication, I think these are around 45, no, this I think they are just 6 replication. You have basically the same quantity of the RD2 if you are working well. If you are not working well, you can have some displacement of the signal, but it's not important, because it's important because you are not working very precisely, but you can have also a good results because theoretically your housekeeping gene is moving with the other one. It's this is difficult to understand like this without example. I will explain something later. So go on. Okay, you see now we have also the signal for the RD5. For example, you see now it's very clear that you have a couple of different signals for the RD5, but housekeeping gene is the same. So you can actually say that and go on. It is actually not so easy to identify. It's one of the most difficult to have. And go on, the RD3, and then also this is what I want to show you. This is the RD4. In this case, you can see that at least one sample is here. Okay, you can see this. it's very difficult without, okay, computer graphic and all the results in numbers. But actually, only by view you can understand that with the same quantity of housekeeping gene, you can have different quantity of expression of the RD4. This is very useful when you have, for example, treatment, and you want to compare if some receptor or some gene overexpress or go down during some treatment. So you have housekeeping gene is stable, and you have more or less expression. I know that this, how can I say, this presentation, it's very complicated by like this. I don't want to go very deep in all of this because you need to see and you need to work in this for understanding better, especially for understanding the results. It's very important that you have a lot of practice in this, and trust me, it's not so easy have this kind of line. I'm showing you one of the best that we have. We can have a lot of problems if we go very fast. For example, in the other one that I show you, this one, sorry, but too much, ecco. Okay, an expert eye, for example, can see very easily that this kind of amplification actually is not so good because it's not linear. There are a lot of problems during, for example, the end of the reaction. And this is good in any case. Also, you can have not amplification or too much amplification. It's okay. All the time you must have a lot of practice in this. But the message for this presentation is that I told you, knowledge is consciousness. And basically, if you are using this kind of technique, you need to understand it deeply. And for do this, you must work a lot, you know that. And of course, constantly form yourself in this. I would like to say thank you to you all for your kind attention. And now that's it.